Hey, welcome to Bio 100 again. We're going to kind of pick up where we left off yesterday. We were talking about animal diversity and some of the major phyla that we have there. Roundworms, sea urchins, of course the vertebrates, right? And then some of my personal favorites, right? The arthropods. Some are highly edible, others are always beautiful. So something really cool to think about in terms of animal diversity. And I realize a lot of you are probably like, wait, I didn't know insects were animals. Well, yeah, insects are animals. We just don't think of them that way a lot of times. And actually, I have a pretty funny story. When my daughter was in fourth grade or fifth grade, I think she had to do a report on a quote unquote animal. And my daughter raised her hand and said, I want to do it on dragonflies. And the teacher came back and said, oh no, you need to do it on an animal. So you can imagine my wife had to restrain me so that I didn't walk in there and give that poor elementary school teacher a lecture on what is and is not an animal. And that kind of fits into where we're headed next. So when we think about the diversity of life, right, specifically when we think about animals, so vertebrates only account for about 4% of all animals, where invertebrates are much more numerous and diverse. They account for the rest, the 96% of animal life. So many invertebrates, they produce compounds that are found nowhere else on Earth. And these compounds can be, have been looked at for usages in medicine, agriculture, etc. But real quick, before we move on, I think a lot of you, if I challenged you to like draw a picture of all the animals that you could think of on Earth, a lot of you would probably draw something like this, which is pretty fair. This is what we're taught animal diversity looks like, right? That you're going to throw in some small things, some beetles, right? Some grasshoppers, dragonfly maybe, and then you're going to be able to draw a whole slew of larger vertebrate animals. But in reality, if we look at the diversity of animals on Earth, this beetle and if all of these animals are actually drawn to scale, what you see right away is that vertebrate animals are represented here by this elephant are much less diverse by beetles here. I guess this would probably be insects here, right? Which is this beetle overall. And in comparison, fungi are more diverse than vertebrate animals. Mollusks are more diverse than vertebrate animals. Bacteria are clearly more diverse than reptiles or amphibians or flatworms. And I guess this isn't vertebrate animals. I apologize. This would be mammals in general. And then you would have mites that are hyperdiverse, worms, other animals. So this is kind of interesting to look at this. And here's plants. Okay, so plants are somewhat in the ballpark of diversity of insects in general, but this is a really interesting way to think about all these and to actually see an image that shows you to scale the diversity of animals on Earth. And there's some of the highlights that we just talked about. Okay, so what's really interesting here is when we look at the diversity of life, right? I've talked about how animals are used for humans, and here are some examples here. So you have this ant that has a venom, and that venom is a potential source of arthritis treatment. You have jellyfish that fluoresce. Remember, we talked about this in the bioengineering section of our course, where there's these fluorescent proteins that are useful for labeling organisms, and they've actually been able to transfer some of these proteins from things like fireflies and bioluminescent jellyfish and put them into cats. So here's a cat that bioluminesces orange-red, and here's a cat that bioluminesces green. And these are used to study whether these cats have overcome certain sicknesses and or diseases. And then we have the horseshoe crabs. So these are a source of blood protein that's used to test for pathogens in humans. And what's interesting about these horseshoe crabs is that there's a whole system. So you'll go and there'll be this swamp, and they'll be right on the edge of this swamp. There'll be this like medical looking storage building on the edge of this swamp. And if you're there at the right time, you'll see what is literally a bunch of hillbillies coming in canoes full of these horseshoe crabs. And what will happen is lab techs will come out, take the crabs, they'll literally throw them on a spike, and they'll bleed the horseshoe crab a certain amount, and then they'll take the blood and they'll push it through to whatever they need to help create kits to detect human pathogens. And then the horseshoe crab, once they're done taking some of its blood, they will throw it back out into the swamp, and the horseshoe crab is not unharmed, but it's not a lethal thing, that they can still go on living. Pretty interesting to think about. Okay, so I want you to hit the pause button here and spend a significant amount of time on this. Okay, so there's the instructions here on the right. There's some hints. You need to be able to pretend that you're working as a field biologist, and you need to identify these different classification schemes of life, and you use all of these examples and these characteristics to be able to identify how you would differentiate between these different groups. Okay, so the affinities of all the beings of the same class have sometimes been represented by a great tree. I believe it has been with the great tree of life, which covers the surface with its ever-branching and beautiful ramifications. So this is Darwin. One of Darwin's major thoughts is that he started to put things together into this tree of life, where he essentially had different branches for different parts of life, but also this idea of common ancestry. And so as a result, it's exceptionally important to be able to understand what information trees are conveying. And this is more of a stylized kind of drawing of how trees look and go, of how trees could be presented. But what we're going to do is we're going to learn how to do a little bit of tree thinking. So scientists suggest that organisms should be classified based on evolutionary relationships. So remember, we had our kingdoms, phylums, class, family, right? Phylum, order, family, a class, order, family. So when you're looking at these, if we're going to group things according to their evolutionary relationships, this requires comparing things like DNA and morphology. So you can think of morphology as like anatomy, comparing the bones between between humans and birds and frogs, right? That would be a morphological comparison, or using morphology to create a phylogeny or a phylogenetic tree. So DNA and morphology of closely related organisms should be more similar than DNA of distantly related organisms, okay? So DNA and morphology, right? The DNA and anatomy should be more similar in species that are closely related than the DNA or morphology of species that are distantly related. So here, let's give this a shot. So, by reference to the above tree, which of the following is an accurate statement of relationship? And I want you to hit the pause button and try and work this out on your own. Okay, so here we go. A green alga is more closely related to a red alga than to a moss. Okay, so this green alga is more closely related to the red alga than to the moss. Okay, a green alga is more closely related to a moss than a red alga. So a green alga is more closely related to a moss than a red alga. Okay, and then we have a green alga is equally related to a red alga and a moss. Okay, a green alga is related to red alga, but it is not related to a moss. So what's the proper answer here? The answer is B, okay, that a green alga is more closely related to a moss than a red alga. Well, some of you might be sitting there thinking, well, that makes no sense. It's equally distant to the two. Well, but you have to think in terms of common ancestor. Where's the common ancestor? Is the common ancestor here at Y or X? So what we're saying is that the red alga, due to common ancestor Y, is equally related to the green alga, moss, and pine. And so, but it's not more closely related to any one of those. So if you're going to take a really hard look at this and you use this as the ancestor, this really helps you get to answer B. Okay, so what we're saying here is that the green alga shares a common ancestor with both moss and pine. Thus, it is equally closely related to both moss and pine. Okay, so give this a read if that doesn't quite make sense. It should. All right, here's the next question. Which of the trees below is false given the larger phylogeny above? So what you need to do is you need to take this tree, and it will be in a simplified form here, meaning that they've removed some of these species, but the same overall pattern of the tree is here, and you need to find which one of these trees is false given this pattern and it being simplified. So can you figure it out? Hit the pause button if you can. Okay, so here's the answer. So D is the correct answer. So D shows yeast being more closely related to plants than it is to animals. The true phylogeny shows that yeast is more closely related to humans than any of the plants. Okay, so that should make sense to you. As we trim things down, this is the only one here that doesn't really make sense. Okay, if you were to add a shrew right here to the phylogeny shown above, where would its lineage attach to the rest of the tree? Okay, so what we're saying is if you had to add a branch that represented this species on this tree, where would it go and why? Hit the pause button, think about it. Okay, hopefully you took some time. It would go here at G. Do you know why? Well, it so happens that this is a mammal. It has fur, it has mammary glands, and so we are mammals as well, so it would sit right here at point G. All right, so if you were to add the pictured organism below to the phylogeny above, where would its lineage attach to the rest of the tree? Now this is a little tricky one. You need to think about this really carefully and look at this really hard. Okay? Where would you put it? So I'll tell you from past experience, hopefully. So hit the pause button, think about it. So I'll tell you from past experience, the vast majority of my classes put this lineage right here with the snake. And that's not necessarily incorrect. However, what if we get some DNA evidence? So here is the lizard, here is the snake, and here is the pictured organism, this one down here. Where do you think it goes now? And you should be able to walk across, to look at here, to look at these, and walk across and be able to see that this is actually a lizard without legs. And if you look really close, there's actually a reduced set of legs right here that don't poke through the skin except for a couple of claws in some species. And there's actually a head here. If you're a biologist and you've looked at these quite a bit, you see right off the bat this is actually just a lizard without legs. And so if we come here and look, you can see that the lizard and the snake have a similarity, but there are more similarities between the lizard and the pictured organism, which is a legless lizard, than there are between the pictured organism and the snake, or the lizard and the snake. And so you know right away that it goes right there. Okay, so we've done a lot of we've talked a little bit about tree thinking, classification, and if you look at where you are, if this represents only about 3,000 species on Earth with humans, you're sitting right here, and you can see it. It says Homo sapiens, sitting right there. One of the cool things about BYU is we actually have an international reputation as being well known for creating phylogenies. These are, we've talked about these a little bit. It's what we were doing in the tree thinking. We create these phylogenetic trees, and we go in and sequence really deeply different parts of the tree of life. So this is the crustacean tree of life. So this is the branch that represents all crustaceans. BYU has been funded to do this in the past. We've also been funded to do the dragonfly tree of life. We've actually been funded to do this twice. We did an initial effort starting in 2012, and we were just funded again in 2020 to do a much larger, more extensive work with dragonflies. And I should say as well, dragonflies just have this really killer fossil record. if you take this fossil and you hold it in your hand and you move it in the light and you look at this area right here, you actually see that there is metallic coloration preserved in this fossil. Not only that, but the striation here, everything about this fossil is just exceptional. You can see the striation in the muscle, their cuticle, and so we have been funded to tie together both the fossil and the extant tree of life. And there are a bunch of fossils here, so we've begun on this. We're trying to work on it. We also study their visual systems. So as part of this proposal, one of the things that we'll be doing is looking at the evolution of their visual systems. Now, dragonflies are fascinating in that their visual systems differ quite a bit. So here's how they differ. So in humans, you have 3 proteins in your eye that allow you to see color. You have one that allows you to see blue, and if you're not colorblind, you have one that allows you to see green and kind of out towards the red part of the spectrum. Now, if you're colorblind, you just have one peak here, and you can't really tell the difference between your greens, reds, browns, etc. And that's because you don't have 2 opsins out there. That's what these proteins are called, opsins. Now, insects also have 3, but they're distributed differently in that they have one opsin out here, this protein that allows them to see light in the ultraviolet range of the visible light spectrum. Blue, pretty close to our own, and then out here in the green as well, in the red-green as well. So what you're looking at is you have a butterfly like this, and we would see it like this, but insects would actually see it closer to something like this in terms of color. Now, that's not exactly true because humans see in much more detail than insects do. So this is probably closer to what an insect would see. It would see the ultraviolet light on the wing here, but it would be almost more of a pixelated form than what humans are seeing. So how has color vision in dragonflies evolved? Well, one of the cool things we're able to do is we can go in and we can take the eyes from a dragonfly and we can sequence the genes that are expressed in those eyes. And we can get an idea of what's going on in terms of dragonfly visual color vision evolution. And one of the things you may notice here is that the ultraviolet opsins are not very diverse in dragonflies. As a matter of fact, every species we looked at only has 1 copy, but they have an expansion in the blue here and a massive expansion in the red. Red is just represented by the long wavelength, which allows them to see in the green, yellow, orange, red portion of the visible light spectrum. So when we look at the patterns of evolution across these different opsin classes, ultraviolet, blue, and long wavelength, one of the things we notice is that the amount of natural selection happening across these different types of opsins is heavily relaxed in dragonflies. And because it is relaxed, it allows for many more copies to be present, as we see here, whereas in these other groups, selection is cranking down really tightly on both blue and UV opsins, and that's why there are not as many copies present in either one of these. But ultraviolet has the highest level of selection, blue has the next highest level of selection, and then the red here is pretty relaxed, and we can see the same thing here. Here's the ultraviolet, right, and this is the amount of selection there. This is the blue and the amount of selection there, and then the amount of selection here, represented in green for the long wavelength opsins. Some other cool things we've been able to do, we've done the firefly tree of life has been funded here, and here's really exciting. We can go in and look at the evolutionary origins of bioluminescence. How many times did fireflies evolve bioluminescence? To support a lot of this research, we actually have the largest insect specimen tissue collection in the world. Dr. Whiting here at BYU has been building it for 25 years. It represents all insect orders, about 700 families, and 1.5 million curated specimens. And all continents are represented except Antarctica. We also have the largest tissue database in the world for both DNA and RNA. You guys know what these are now, right? And there are about almost all families are present here now. This needs to be updated. And there are about 300 genera and 900 species in the collection. And this was started by Dr. Whiting, and I've been carrying it out since. And here's just some of the diversity that you see. Here's a dragonfly here, and here is a damselfly. incredible damselfly. These guys feed on orb weaver spiders in Central and South America. And that's where we'll leave it today. Hopefully that was informative. You guys got a little bit out of it. And looking forward to headed into the last stretch here for the class.